Welcome to EPG Pathshala lecture series on data analytics. In this session, we will be discussing about the rest of the topics related with clustering. The learning objectives of today's session are: know about clustering algorithms for arbitrary shaped clusters, and algorithms like CURE, CLICK and PROCLUS algorithm that cluster arbitrary shaped clusters will be discussed as part of this session. Now, let us start with the CURE algorithm. CURE algorithm in some way or other looks like an extension for K-means algorithm, but here in this scenario CURE algorithm works on arbitrary shaped clusters. We will be discussing in this order. First, we will discuss about the phases of CURE algorithm, then an introduction to CLICK algorithm and the steps involved in it. Finally, we will be discussing about PROCLUS algorithm and its working. Normally, when we cluster, and whatever algorithm that we discussed so far are related to clusters that are either aligned to a specific axis or they are circular or elliptical in nature. But in real scenarios, we may end up with clusters that are not related to any shape. In other words, they are arbitrary shaped. Whenever we take this BFR, which is an extension of the K-means algorithm, it assumes that the clusters are normally distributed in each dimension and the axes are fixed. So, what happens is, when a particular cluster which is not aligned to a specific axis will be discarded, but this need not be correct in all scenarios. CURE algorithm is one algorithm which takes care of clusters which are arbitrary in shape. It makes use of the assumption like it uses Euclidean distance, it allows clusters of any shape, and it collects representative points to represent clusters. When you take a look at the clusters given in this slide, initially we were discussing about clusters which were looking either in a circular shape or we were looking at clusters that were elliptical in nature. When you represent this cluster, they are aligned either to the x axis or to the y axis, but when you take a look at this clusters, they are few of the clusters are aligned to x axis, but say this particular cluster is not aligned to either the x axis or to the y axis. Therefore, this cluster sometimes may be ignored. Take a look at this cluster, the diagram, the cluster diagram. Here the clusters are not associated to any of the shapes that we have discussed earlier, like the circle or the elliptical shapes. It is arbitrary in nature. So, when you want to work on such arbitrary shaped clusters, then we require an algorithm like CURE. Now, let us understand the working of CURE algorithm. CURE actually works in two phases. As I already mentioned, CURE is clustering using representatives, that is, it will be identifying representatives for each and every cluster and then using it for further fine tuning those clusters. So, the two phases of this CURE algorithm goes like this. In phase 1, it actually selects random samples of points that may be fitted into the main memory. So, in other words, you will have to choose points which is sample and the size of the sample is chosen such that it gets accommodated into the main memory. So, what it does is it will create an initial cluster, that is, it will make use of any hierarchical technique and then it will group the nearest points or the clusters. When it wants to do so, it has to come up with representative points. So, having decided how many number of clusters you require, you will have to choose that many number of representatives. So, for each cluster we will have to pick a sample of points and it has to be dispersed as possible, and the sample that you pick should have representatives. These representatives will then be moved towards the centroid, that is, they will be dragged towards the center. So, to what extent these representative points will be dragged towards the center is by 20%. Let us understand this with an example. Now, consider set of points represented with notation E and H over here. We find say two clusters, one comprising of similar data points and another one also comprising of similar data points. H and E respectively. There is another region in the cluster that has dispersion of data points in both the scenarios. One is the H case, another one is the E case. Now, in such scenarios, when you apply this Cure algorithm, you will be choosing some representatives, and while choosing the representatives, we will have to be careful. We should see to that it is separated by a larger distance. So, in this example, we find 4 points are selected and these 4 points are far away from each other. In other words, they are remote points. Then the next step is you will have to pull these 4 points towards the center. So, by 20% extent, you actually shrink these points towards the center. So, what happens is these representative points actually come close towards the center. Once this phase is over, in the second phase what happens is we will have to once again rescan the whole data set and take each and every data point P and then understand whether it can be accommodated into the existing cluster or not. Now, when I talk about a data point and identifying whether it can be accommodated or not, we should think about the closeness property. But closeness here varies from one type of analysis to another type of analysis. As we discussed in the previous session, there are many ways by which you can represent closeness. The normal definition for closest is you try to identify a point P that is very near to the centroid, and if it is very near to the centroid, then you are going to assign that particular representative to the cluster. This is what is happening as part of the phase 2 of Cure algorithm. So, when you try to make use of the representatives, shrink them and then merge towards the center and identify certain points that are close to this center and then call them as cluster is what is Cure is doing. Having understood about the Cure algorithm, now let us analyze another algorithm called CLIC. CLIC is actually a density based clustering. So, what exactly happens in CLIC is it has to cluster based on say a criteria. The criteria normally will be a local cluster criterion. It looks in for densely connected points. What are the major features? It actually discovers clusters of arbitrary shape. It is one type of density based clustering algorithm, and it also takes care of noises. So, even if you have a noisy data, it clusters very effectively. It performs only one scan and it needs density parameters for the purpose of terminating the algorithm. There are many algorithms that are based on density like DBScan, Optics, DenClue and CLIC, to mention a few. We will be working in detail about the last algorithm CLIC, which was proposed by Agarwal et al. In 1998. So, the full form of CLIC algorithm is clustering in quest. So, this was proposed as mentioned earlier like Agarwal, Gherke, Ganopoulos and Raghavan in 1998. This particular algorithm automatically identifies subspaces in high dimensional data space and it uses such dimensions for the purpose of deriving the clusters. So, it partitions the data. For each and every partitions, it makes use of the dimensions related to the partitions and then it looks in for densely connected data points which will be put together for the purpose of forming cluster. So, finally, a cluster which is maximal set of connected dense units within a subspace is what is going to be the output for CLIC algorithm. Let us understand this CLIC in detail by taking a look at various steps involved in CLIC algorithm. So, the first step is you need to partition the input data space and you will have to find the number of points that lie inside each cell of the partition. Now, having partitioned the data and understanding about the data points inside those partitions, next we should identify subspaces that contain clusters using a priori principle. Now, when I say identify clusters, what is that we are trying to do is we are trying to determine dense units in all these subspaces and then we try to identify such connected dense spaces so that it forms a single cluster. So, ultimately we are trying to identify connected dense units in all subspaces for the purpose of generating clusters. When we are trying to generate the clusters, we also try to generate a minimal description for these clusters. So, the minimal description is associated to a maximal region that covers a cluster of connected dense units for each and every cluster, and we also determine the minimal cover for each cluster. So, the minimal and maximal region associated to a particular cluster provides a description about that particular cluster. So, what are the strengths and weaknesses of click algorithm? The major strength is it automatically identifies all the subspaces in a high dimensionality environment, and this is what is very essential when you are trying to deal with a voluminous data set with very large dimensions, because in subspaces only you can find densely packed data, and these densely packed data really helps in forming the clusters. Another advantage is it is insensitive to the order in which the data points are provided as input to the algorithm, because in certain algorithms based on the input, the order in which the input is provided, the cluster and the output of the clustering technique may vary, but this is not so in case of click algorithm. The third advantage is it scales linearly with respect to the size of the input. There are other algorithms that we discussed earlier, they scale exponentially or in certain scenario it scales polynomially, but in case of click algorithm it is scaling very effectively with respect to very large inputs. These are the three major advantages of click algorithm. Apart from this, there is also a downside related to click algorithm. Whenever we are trying to work with respect to clusters that are formed by densely connected data points, the accuracy of the data points may be dragged or it can even be degraded because of the simplicity of the method. The reason is every time you are trying to identify densely connected data points, sometimes there may be points that lie outside and these points may not result in identifying connected units. Therefore, the accuracy of the cluster that we define may be wrong. Having understood the steps, advantages and drawbacks of click algorithm, now let us take another algorithm called Proclus algorithm, which is also known as projected clustering algorithm. So, Proclus means it is projected clustering. What exactly this algorithm does? The algorithm actually finds out clusters and the dimensions that are corresponding to these clusters, because when we are trying to project or visualize the clusters finally, what is essential is the dimensions that are relevant to these clusters. Finding dimensions that are relevant to any cluster is going to be an additional task, but here in this scenario Proclus algorithm does it implicitly. It also identifies the outliers and then the outliers are kept away from the normal clusters. So, outliers are nothing but points that do not cluster well or points that does not part which is not part of any of the cluster. What are the inputs and outputs to Proclus algorithm? The input is going to be set of data points which is going to be very large in number. We are going to also mention how many number of clusters we require as the output, the average number of dimensions for each and every cluster which is denoted by L. If these three are provided as an input, the output for this algorithm will be the clusters formed and their respective sub dimensions. Those sub dimensions are going to be very, very small when compared with the original sub dimension L which was considered. Let us understand what are the various steps involved in Proclus algorithm. Proclus algorithm works in three phases. The first phase is initialization phase, the second phase is iterative phase and the third phase is refinement phase. In the initialization phase, it works towards identifying centroids. In the second phase, that is iterative phase, it looks for correct centroids, and in the third phase it looks for outliers and then eliminate them. Now, let us take in-depth look into these three phases. We will start with the initialization phase. In the initialization phase, we choose a sample set of data points randomly and then we choose a set of data point which is probably the medoids of the cluster. So, the medoid here in the sense it is a middle value which represents the sample which you have collected. So, how many number of medoids we normally select, it depends on the number of clusters. The medoids here in the sense are data points which is nearest to the center of the cluster. So, it in other words acts as a representative for the set of data points or for the clusters. For example, here in this diagram you may find various data points represented using cross. The center of this cluster is indicated by the blue shaded plus sign, and there is a data point indicated using brown color. This data point is considered to be the medoid because it is this data point which is very nearest to the center of the cluster. Likewise, we will have to identify many number of such medoids. During the initialization phase, as already mentioned, we initially select many data points and then we choose at random some set of sample. This sample data size will be small when compared with the original data. Having chosen sample points, next we will have to choose set of points which also includes the medoids. The medoids are represented by means of the notation M. Now, choosing the medoids is done by means of a greedy approach. Finally, during the iterative phase, we verify whether the medoids that you have chosen are the correct medoids or not. Now, when you take a look at this process, we initially start with a very large data, then we finally narrow down to a data whose size is k, which is very small when compared with the original data. Having understood the initialization phase, we should also know how the medoids are selected. I told medoids are selected based on a greedy approach. So, what is that you need to do is, while selecting the medoids, you should see to that the medoids are separated far apart. Now, for this you will have to start with a random point. Say for example, if there are 5 data points represented as A, B, C, D and E, the matrix given over here highlights the distances of these data points with each other. Now, what is that I can do is, I can randomly pick a point A. Then if this point is chosen as a medoid, the next point which I choose must be based on a greedy approach. So, the greedy approach here is identifying a data point which is very far when compared with respect to the data point that I have chosen randomly. So, here from this matrix you can find, that is in the first row, that represents the data point A and the distances of this data point with respect to the rest of the data points is what is selected and highlighted here. You find point A is very far with respect to point E. This can be seen based on the value, the distance value. So, A is separated at a distance of 7 with respect to E, with respect to D it is at a distance of 6, with respect to C at a distance of 3 and with respect to B at a distance of 1. So, the next element that will be chosen as medoid will be E. Now, this is very important. The reason is, only when the medoids are chosen and they are far apart, the clusters that we form will be correct, they also will be separated. So, as part of this greedy approach, we will be choosing only those data points that are very far away, and since we are using a distance metric that is greedy in nature, we say it is a greedy algorithm. So, during the iterative phase, we have to evaluate the medoids that we have chosen. So, what we can do is, the medoids that were chosen as part of the initialization phase indicated by M will have to be identified whether it is the best medoid or not. So, what you can do is, you can randomly find the set of points M, say a point that you consider can be indicated using the notation M current, and replace the bad medoids from other points in M if necessary. Let us understand how this is done in detail. So, for medoids, following will be considered for each and every dimension. So, what is that you need to do is, when you select a medoid, you will have to find all the dimensions related to the medoid and then assign data points to this medoid. Once you assign data points to this medoid, it looks like a cluster. What is that we should do next is evaluate the cluster which is formed, and when you find that medoid does not provide a good cluster, then you will have to mark that particular medoid as a bad medoid, and then you will have to replace it by means of finding another best medoid, and this procedure need to be repeated until we got a satisfied result. So, during the iterative phase, how to find the dimensions? So, for each medoid mi, we will have to choose d, which is going to be the nearest distance to the other medoid, and all the data points that is within this distance will be assigned to the given medoid. Say for example, in this diagram, assume there are 3 medoids A, B and C. Now, we take point A and then we identify among the rest of the 2 medoids B and C, which one is the nearest. C is nearest. Now, we fix a distance from A to C as a minimal distance and identify what are the data points that fall within this neighborhood, which is defined by this minimal distance del. So, we find 4 data points falling within this, and hence we say that A seems to be a good medoid when compared with B. Having identified the medoid, the points which are very close to the medoids, then we will have to calculate the average distance of each and every point with respect to the medoid. So, once we have calculated the medoid A, next you will have to calculate the distance of all the points within this cluster, which is going to be the distance with respect to the center of the cluster. Having calculated this distance, next we can calculate the mean standard deviation along the dimension xij. Then you will have to calculate the variance z with respect to the dimension xij. Then you will have to choose such way the dimensions which are most negative with respect to the given variance zij. So, normally we choose the least 2 negatively applicable dimensions. So, in this phase we are trying to identify those dimensions that are very close to the given set of points and the given medoid. Then in the iterative phase, what is that you need to do is, having identified the data points, the respective dimensions, then you can make use of Manhattan distance to identify whether a data point can be associated to the given dimension or not. When we talk about Manhattan distance is actually a segmental distance between 2 points, say for example x1 and x2 for a given dimension, and it is defined as given in this equation. So, distance between the 2 points x1 x2 is summation of the difference between these 2 points divided by the total distance. Having identified these points, medoids and the respective dimensions, next you will have to evaluate. So, while evaluating, what is that you need to do is, you need to make use of these 2 equations and calculate whether a data point associated to a given dimension is within a given range or not. Having evaluated, next what is that you should do is, you should try to compare, say the previous case where you have considered one medoid and the new case where a new medoid has come into picture. In case the error with respect to the new medoid is less, then you go ahead with the new medoid and ignore the previous medoid. Then the last step in Proclus algorithm is, you will have to redo the iterative process until the best medoids are chosen. Medoids are so important because when you get the final cluster, the final clusters must be far apart, that is separated. Only when you choose the correct medoid, the clusters that you get will be separated by say a larger distance, else you may find overlapping clusters or you may find wrong data points in the wrong cluster. So, during the iterative phase, many iterations may come into picture and many medoids will be chosen, few will be evaluated and then ignored if they do not form the best medoid. So, as a result of the iterative phase, finally you may end up with some set of best medoids, and these medoids are evaluated based on the Manhattan distance metric. Now, in order to improve the quality, why we need to improve the quality at this point is, see, there may be certain points which though close to a given centroid sometimes may not fall within the range. So, I may have an outlier. Whenever there is an outlier, the outliers need to be identified and eliminated. So, how to handle the outlier? We were discussing about the Manhattan segmental distance. So, for each and every dimension and the corresponding midpoint. So, when you choose a midpoint, you will also have a set of dimensions related to the midpoint. Now, we will have to find the smallest Manhattan distance, Manhattan segmental distance to any of the other midpoints, and in case if this Manhattan segmental distance is minimal, then those particular data points will be retained and the rest of the data points will be ignored. So, what happens is, you try to calculate the Manhattan segmental distance, identify what are those midpoints with respect to a given dimension have very minimal Manhattan distance, and only those points or midpoints will be retained and the rest of the data points will be ignored. So, finally, what happens is, we start with set of data points which is a sample, we select certain midpoints and then we evaluate whether the midpoints that we have chosen are the correct midpoints. Having chosen the correct midpoints, next we will have to identify the sub dimensions related to those midpoints. Then we will have to identify whether the sub dimensions are correct to the given midpoint. So, we do next level of evaluation, and finally, we may have certain outliers which need to be eliminated. For eliminating the outlier, you need to take into consideration Manhattan segmental distance between various midpoint points. So, those midpoints that share minimum Manhattan distance will be retained and the rest will be ignored. So, what happens finally in the refinement phases, we make use of the minimal Manhattan distance del I, and in case if a particular midpoint M I is within the sphere of influence, then we consider that midpoint, else we treat that particular point to be an outlier and then we ignore it. So, finally, what happens is, we end up with a set of clusters and also we identify outliers, and those outliers will be ignored as part of the 3 phases of Proclus algorithm. So, among the 3 algorithms that we discussed, the Cure algorithm, the Click algorithm and then the Proclus algorithm, we find Proclus algorithm working very effectively because there are multi levels of evaluation. So, to summarize, algorithms for deriving clusters of arbitrary shapes actually require different distance metrics and different approaches. The reason is, when we are aligning or identifying clusters that are aligned to a specific axis, the distance metric that we choose may be fundamental metrics, but when we go further for analyzing arbitrary shaped clusters, we may require more heuristics for the purpose of identifying such clusters, and finding subspaces and relevant dimensions become very necessary when we are dealing with very large data sets. So, to summarize, when we are discussing clustering techniques, there are many clustering techniques. Each and every type of clustering technique deals with a specific type of output. So, you need to be very clear on what is the requirement, what type of cluster is essential or what type of cluster is required as an output. Accordingly, input must be transformed, it must be provided to the algorithm. By choosing relevant distance metrics, you will have to derive the clusters. Only then the output of any clustering technique will be correct. So, you will have to have a proper mapping between the requirement and the choosing of algorithm. Thank you.